talk, so I won't keep you long away from your lunch. But again, tells you a point that always question what you read if the evidence does not stack up. So all cornea surgeons know that the Descemet's membrane always rolls with the endothelial cell outside. So when we do a DMEC procedure, we take the Descemet's membrane out, the endothelium is outside, the Descemet's membrane inside, and the same happens with PDEC tissue. So when you make a bubble, the type 1 bubble, which has the pre-Descemet's layer, Descemet's membrane endothelium, you can cut it out or you can refine it, and then you can get this piece of tissue. This one scrolls less than the DMEC tissue, but it also scrolls with the endothelial cells outside. This scrolling is absolutely crucial for us to get the right orientation when we open it in the eye. But nobody has been able to clearly explain why it scrolls in that manner. So if you look at this montage here, this is PDEC tissue, 4 different samples. And from this tissue, if you very carefully peel away the Descemet's from the pre-Descemet's layer and you lay them side by side, the pre-Descemet's layer scrolls less, but the Descemet's membrane scrolls much more, and this one is in between the two, as you see in all. So most of the scrolling of PDEC tissue is also imparted by the Descemet's membrane, because the pre-Descemet's layer on its own scrolls less than the PDEC tissue does. So here you can see the statistically different scrolling of PDEC tissue, DMEC tissue, and the pre-Descemet's layer, Dua's layer on its own. So everybody has said it is because of the elastin in the Descemet's membrane. Now that's true. Descemet's membrane has elastin. So if you measure the elastin of the different parts of the cornea, the pre-Descemet's layer and the trabecular meshwork have a very high concentration of elastin. In fact, the pre-Descemet's layer has more elastin than the Descemet's membrane. And this has also been shown by on-face scanning electron micrography by somebody else, another group, Keith Meek's group in Cardiff, and they've shown there is a lot more elastin in the pre-Descemet's layer. Now look at this staining. All that is green is elastin. So this is the pre-Descemet's layer, has elastin all over it, all through the tissue. This is Descemet's membrane from here to here. And are you seeing my, no, you're not seeing my, sorry, my arrow. So once again, this is the pre-Descemet's layer, and all of it is elastin, and this is the Descemet's membrane from here to here, but the elastin is only concentrated in the band in the anterior 1/5 or 1/6 of the membrane, corresponding to the banded zone of the Descemet's membrane. Here what you can see is this is PDEC tissue. So the pre-Descemet's layer is there. There's the band of the Descemet's membrane, and here you can say where half of the pre-Descemet's layer has been taken away. So you can see that's PDEC tissue here, and that's DMEC tissue. So that's the difference. Now, if you take a rubber band and you pull it and you leave it, so then you'll find it'll go like that, it'll come back. But if you take a rubber band and you pull it and you stick it to a piece of paper, when it comes back, the paper will roll that way. And that's what exactly happens here. The elastin distribution is what determines the direction of the scroll. Because the elastin is here, it rolls that way so that the endothelium is outside. It's like the rubber band stuck to a piece of paper. And if you look at the whole cornea section, that is stroma, that's a keratocyte, you can see the pre-Descemet's layer very clearly different in its elastin content. That's the interfacial zone where it's dark, and that's the Descemet's membrane with the band. So it's not just in the separated tissues, but it is also in the whole cornea that you see this unique distribution of elastin in the pre-Descemet's membrane, pre-Descemet's layer and the Descemet's membrane. Now, the proof is in this diagram here, this montage. That is Descemet's membrane, nicely scrolled, endothelial cells outside. And if you put some elastase enzyme and leave it in front of your eyes, very slowly the whole thing opens up. The scroll is gone. And then if you make a section of this that is unscrolled, you will find the whole elastin band is completely degraded and fragmented compared to the control where it is nice, smooth and uniform. And interestingly, the lens capsule also behaves exactly the same with the epithelial cells on the outside when it scrolls, and it also has a similar distribution of elastin. Now, in the books, it has been written that this scrolling occurs because of the swollen endothelial cells of the demec tissue. So by the time you take it from culture, from the eye bank, and you take out the Descemets, the endothelial cells are stressed, they're swollen, and they push it to roll like that. Now, that's what's written, but if you take a sheet with cells on it, and you treat lightly with Dispace and get rid of all the endothelial cell, it will still scroll in the same direction, telling you that the swollen endothelial cells, which the textbook says, is actually got nothing to do. So how these people came to that conclusion, just because they thought it might be, but there was no evidence. And so that's what happens. This is the pre-Descemet's layer. It acts like a rubber band. This is Descemet's membrane. It scrolls that way, and that's the this is the demec tissue, and this is the PDEC tissue, where the PDL is there. So most of the scrolling is with that of the Descemet's membrane. Now, the question that finally asks you is that God did not put elastin on the Descemet's membrane so that we could do DMEC. It is there for a physiological purpose. And what might that physiological purpose be? So you can imagine the cornea is convex like that and concave internally. The Descemet's membrane has to be applied to the cornea like that, right? Now, Descemet's membrane anterior surface, endothelial cells here. Now, what keeps the Descemet's membrane always attached to the back of the cornea is this elasticity, which makes it want to go like this. It wants to go like that, therefore it's stuck to that tissue there, and it can't go like that, but it keep it compressed. And there is nothing holding them, nothing physical between Descemet's membrane and the pre-Descemet's layer. Yet, that apposition is so important for the endothelial cells to work and pump out the water from the cornea. So this elastin is what is allowing it to hold its position and without any physical attachment. So that's the physiological role. And this paper, as you saw at the beginning, we have a number of authors, and I'd like to acknowledge them all, and including Dr. Said, who is here in the audience. Thank you very much.